Hello to everybody. Good morning. It's Wednesday morning. It's 10 o'clock, so it's time to have fun in Enjoy Science. I want to thank very much to our speaker today, Jaya Chandra, for her time and being with us. And of course, I want to say thank you to the person who makes this happen, our Liz Kent, Laura Adame, Alison Garth, and also to thanks to Dr. Giulio Draeta for taking good care of this series, and to my friend Alessandra Ferraioli, who is co-hosting with me Enjoy Science. And of course, I want to thank all of you who are coming talk after talk and who enjoy science with us. Without you, the people who are attending from MD Anderson and from all over the world. So without you, the MD Anderson family, this webinar make no sense. And with this, let me to say a few words about Joya, because all of us here in MD Anderson and the people from connection with MD Anderson knows her, because she is, in fact, we are proud of her. She's a product of MD Anderson and University of Texas education. She was a GSBS PhD student, and she had wonderful postdoctoral fellow steps in Karolinska and in Mayo Clinic. So all of us would love to go to Mayo and Karolinska postdocs or collaboration or working with that people. And in fact, now she's running as an associate professor in pediatrics, a wonderful research program in exactly what I think it's a central part of cancer therapeutics, about children with cancer. Because let's be fair, most of us are parents. What can be worse as a child with cancer? So in fact, Joya, her group, and many others are focusing on this very important aspect of cancer, the cancer therapeutics and understanding the cancer mechanism in children. And in fact, she is doing a wonderful job. A lot of papers focusing on the oxidative stress in cancer and how this can be turned against malignant cells. And with this, Joya, thank you for being with us. We are very curious about your talk, and good luck with the presentation. Please take from here the show, run the show. Thank you again.
Thank you so much, George. So if anyone has ever gotten an email from George, there's always these commands: enjoy science, be happy, smile. So thank you, George, for being such a wonderful ambassador of science and of enjoying science, and for kind of coming up and perpetuating this webinar. And I'd also like to thank the CSO's office, Liz, Laura, Dr. Draeta, for kind of giving us a cadence as a community with the Tuesday and Friday morning seminars. With this webinar series, really helps us feel like we're a community, despite the issues with actually seeing each other in person as frequently as we used to. So I'm trying to forward my slides, and I'm having trouble. Oh, here we go. All right, so thank you again, and thank you to everyone for joining. So today I'm going to talk to you about trying to improve outcomes for pediatric cancer patients, as George said. I'm in the Department of Pediatrics Research, where every faculty member here is really striving towards the same goal. And I also have a joint appointment in the Department of Epigenetics and Molecular Carcinogenesis. As you all might know, they're all moving to Houston, and there were a series of excellent talks last Friday, kind of highlighting some of the wonderful basic science that group brings to MD Anderson. So I wanted to just start with some facts about pediatric cancers. And so you may or may not know that the number one cause of death by disease for children in America is cancer. And on the right here, we have an infographic of childhood cancer from 2021, courtesy of a foundation, Alex's Lemonade Stand Foundation, which is actually a parent-run philanthropic organization that funds pediatric oncology research specifically. And so I think it's really striking that these families that lose their children, as George mentioned, this is one of the devastating things that can happen to anyone, find it within themselves to support pediatric cancer research to such a great extent. And in this infographic, you can also see some of the numbers associated with incidence of pediatric cancers in the United States. 43 children per day are diagnosed with cancer under the age of 19. And then in the world, every year, 300,000 new cases, 800 children a day worldwide. So this is not a trivial number of individuals that are being affected. We also are very cognizant of the long-term effects of childhood cancer care. And we know that childhood cancer survivors, as depicted in this infographic, are twice as likely to have chronic health conditions that basically limit their lifespan significantly even after they've been cured of their cancer. So another aspect of pediatric cancer is the burden on society. The fact that when adults are diagnosed with cancer, their average age is 67, and there's approximately 15 years of life lost, whereas children, the average age of a cancer diagnosis is 6, which adds up to over 70 years of life lost. So this is a detriment to society and the contributions that these individuals may have moving forward. And we're very aware of the amazing things that pediatric cancer survivors can give back to society, because our head of pediatrics is actually a pediatric cancer survivor. So Dr. Gorlick was a pediatric cancer patient as a child and now leads a world-renowned laboratory looking at osteosarcoma therapeutics and creating precious PDX samples and models for studying this pediatric cancer. So I mentioned that Dr. Gorlick studies osteosarcoma, which is one of many different pediatric cancer tumor types. And I just wanted to highlight the heterogeneity of various tumor types that strike children. And so these two graphs are basically showing the distribution and incidence of different types of cancers within pediatric populations. On the left, in the bar graphs, you can see that brain tumors are more common, inching above leukemias in more recent years. So this is SEER data from 2018. 54.8% of the cancer diagnoses are brain tumors relative to 50% of leukemias. This is not necessarily the same in all parts of the world. So on the right, this pie graph is showing similar numbers between leukemias and brain tumors in terms of incidence. But as you might notice from the way that the word leukemia is spelled, this is from a British cohort. But in general, leukemias and brain tumors are the most common types of pediatric cancers, but there's also a lot of heterogeneity in different tumor sites, retinoblastoma, renal cancers, liver cancers, bone cancers. So all pediatric cancers are certainly not the same, arising from very different etiologies. This is just another look at the incidence of pediatric cancers, this time divided by age ranges under the age of 19. So for children under the age of 14, ALL and brain tumors are the most frequently seen childhood cancers. But if you go a little older, amongst slightly older patients, patients with between the ages of 15 and 19, these adolescent patients, that distribution changes. And so then the most frequent cancers are no longer ALL and brain tumors, but they are within the top occurring tumors. So outcomes are also very heterogeneous for pediatric cancers. There's often a heralding of pediatric cancers as being cured. The cure rates for pediatric cancers are often really highlighted, and that's largely due to the successes in treatment for acute lymphocytic leukemia. And this graph from a New England Journal of Medicine paper is really highlighting the progress that's been made over the decades in improving outcomes for pediatric acute leukemia patients. But on the right, from SEER data, we can see that the bars that are, these multicolored bars, are all at different levels. And so even if you look at leukemias, lymphoid leukemias, the first set of bars versus other leukemias, there's a big difference in overall survival between ALL and other types of leukemias. And then you can also appreciate from this graphic the impact of race on outcomes. So each colored bar represents different racial ethnic groups. And again, you can see that the bars are not at the same level, and that minorities frequently are seeing worse outcomes than white or Caucasian patients. So certainly a lot of heterogeneity to still understand the molecular basis for, in addition to just disease type. So this is just a different way of looking at the survival rates, the outcomes for pediatric cancers. In the middle of this figure, you have the overall survival for pediatric cancers, which is roughly around 83% at 5 years. And then above this, you have some of the tumor types where outcomes are better. ALL is right around 90% survival. But at the very bottom of this graph, you see two tumors, DIPG, a brain tumor that I'll talk quite a bit about, and acute myelogenous leukemia, a different type of amyloid leukemia for which survival is poor. So these two tumor types are ones that our lab focuses on. And so again, we study both blood cancers as well as brain tumors. And we've also integrated a role for energy balance, as I'll tell you later, into our research. And this is primarily driven by those survival rates that I showed you in the prior slide. So for leukemia patients, we really need strategies that improve existing treatments for subsets of patients, such as AML patients. And we also need ways to improve quality of life into survivorship. That's where the role for energy balance comes in. And then for brain tumors, especially for brain tumors like DIPG, where less than 1% of individuals will survive, we need new and better targets and better drugs. So I'll begin by talking about DIPG, this really lethal type of brain cancer that strikes primarily pediatric populations. DIPG stands for diffuse intrinsic pontine glioma. And this is another infographic from, again, a philanthropic organization run by parents of a child that passed away from DIPG. And so this really lays out some of the stark facts about this disease, the poor survival rate, the age range. Children between 4 to 11 are primarily affected. And so this type of cancer was really revolutionized by sequencing efforts that did whole genome sequencing in adult pediatric high-grade glioma patients relative to pediatric high-grade glioma patients. And one of the major discoveries from those efforts was the discovery that there's these unique mutations that are seen almost exclusively in pediatric high-grade glioma patients. And amongst these mutations are mutations in histone proteins, histone H3, and its variants. So these histone H3.3 or 3.1 mutations are really unique, again, to pediatric high-grade glioma and to DIPG. There's several other unique features of pediatric high-grade gliomas relative to adult high-grade gliomas that, again, have primarily been discovered through sequencing efforts. In our lab studies, some of these epigenetic targets, as well as some of these kinase signaling targets, today I'm really going to focus only on DIPG, where this H3, histone H3 K27M mutation is a defining mutation for pontine gliomas in this class of pediatric high-grade gliomas. And so what are these histone proteins where these mutations are found to occur? Just a reminder or a primer for some of you about what histones are. So histones are integral to the packaging of DNA. Here you can see chromosomes organized into chromatin, and then the nucleosomes, which are composed of DNA wrapped around these histone proteins. These histone proteins have these N-terminal histone tails, which get highly modified by post-translational modifications. So these tags, if you will, can be acetyl tags, methylation tags, on either lysine residues or arginine residues. They can be phosphorylation. There's a lot of different post-translational modifications occurring on these histone tails. We were specifically interested in lysine methylation, and it's important to note that these methylation events are not just putting on a tag and taking off a tag, they're iterative. So there can be a mono, di, or tri, so up to 3 tags, methyl tags on either lysine or arginine residues. So the mutations that have been noted in DIPG patients are again occurring in histone H3. You saw in the previous slide that there's several different histone proteins, and this is just a closer look at the tails of those histone proteins. You can appreciate from this slide that histone H3 is amongst the most modified, has more of these tags on its amino acid residues than the other histone proteins. And in particular, you can see that some of these residues, such as this K or lysine at position 4, or this K or lysine at position 9, can either be acetylated as depicted by the blue tag or methylated as depicted by the red tag. And so the methylation, the red tag, is negotiated by an enzyme that we have become quite interested in called LSD1 or KDM1A. And so LSD1 is lysine-specific demethylase. KDM stands for lysine, the K residue is lysine demethylase, and it was the first lysine demethylase discovered by Yang Shi at Harvard in 2004. This is the domain, the various protein domains of LSD1. It's an amine oxidase, so this is the mechanism by which it actually exerts its catalytic activity, and you can see the amine oxidase domains here. It demethylates mono and di, H3K4 or H3K9, and it's frequently found in these complexes that frequently contain HDAC family members. And our specific interest kind of was stemmed by the fact that it was frequently in complex with other negotiators of histone marks. We were also very intrigued by the ability to pharmacologically inhibit LSD1. And this is because LSD1, as shown in this ribbon structure, shares a lot of structural features with monoamine oxidase B. And monoamine oxidase B has been a target for decades for the treatment of depression. And one of the drugs that's been used to successfully treat depression by targeting monoamine oxidase B is tranylcypromine or Parnate. And this drug was found to also inhibit LSD1, actually by Mingyu Li, who's an associate professor at MD Anderson as well, back when he was a postdoc. So this drug is known to cross the blood-brain barrier because of its efficacy in treating depression. It's an inhibitor of LSD1, and so we were intrigued by the fact that there was a ready-to-go pharmacological tool to inhibit this important enzyme. And so I mentioned to you that H3K4 and H3K9, the lysine residues that LSD1 acts upon, can either be acetylated or methylated, and that some of the complexes that LSD1 is part of, such as the co-rest complex, also include HDAC family members that modulate the acetyl mark. This led us to think about actually targeting these marks simultaneously. And back in 2005, we actually showed that with two different drugs, one of them being tranylcypromine or with HDAC inhibitors, there's synergy and preclinical activity in adult glioblastoma. Since then, Yang Shi, again, who discovered LSD1, actually published pretty recently a very interesting paper using a single drug that has both HDAC inhibitor capacity as well as LSD1 inhibitor capacity to show that differentiation of DIPG was promoted and had potential as a therapeutic. So this is the graphical abstract from that paper. But as this was emerging from 2005 to the end of 2019, the field exploded with new LSD1 inhibitors. And this was mostly because of this backbone of tranylcypromine, which was capitalized upon by several companies to inhibit the catalytic domains of LSD1. So when we initiated our studies, we were really looking at various LSD1 inhibitors that were catalytic inhibitors that had this backbone similar to tranylcypromine. But there are other inhibitors, which are scaffolding inhibitors, such as a compound from Solarius Pharmaceuticals right here in Houston, which is actually in phase 1 trials for pediatric patients now for Ewing's sarcoma. And this agent is somewhat unique in that, again, it's binding to other domains, not just the catalytic domain of LSD1. And so our initial studies kind of looked at all of these pharmacological agents collectively. So we began by looking at some unique cell lines that were actually PDX cell lines, so patient-derived cell lines from patients that had passed away from DIPG, which have been collected and distributed by Michelle Mongi, who's at Stanford. She provided us with 3 different DIPG cell lines, and these are all bearing the histone mutation that kind of defines these pediatric high-grade gliomas. And so with this range of inhibitors, we found that the catalytic inhibitors were showing lower IC50s compared to normal human astrocytes or even adult GBM. So there appears to be some selectivity for single agent LSD1 inhibitors, specifically catalytic inhibitors for DIPG cell lines. So this is intriguing, but we wanted to also understand what these agents are doing kind of more broadly. And so to do this, we went back to LSD1 itself as a drug target and knocked it down in adult GBM cells to understand what genes are controlled by LSD1. And so this is RNA-seq data, generated originally in adult GBMs, where we found that amongst the most highly changed genes were genes that control the immune response. These genes were actually represented quite strongly in a database of pediatric high-grade gliomas. So this is TCGA kind of focusing in on 247 pediatric high-grade glioma cases, and this relationship between LSD1 knockdown and the upregulation of the immune genes was conserved in this data set. So using these inhibitors, this suite of inhibitors that I introduced to you, we then looked to see in the DIPG lines whether the immune genes were actually modulated by these various LSD1 inhibitors. And these are violin plots where each color represents a different LSD1 inhibitor, and each kind of violin represents that collective group of immune genes. And so you can see that in the DIPG4 and DIPG6 cells, for several of the catalytic inhibitors, tranylcypromine or its cousins, there's a significant increase in those immune genes. This is not seen in normal human astrocytes. So not only is there selectivity in terms of cytotoxicity for the LSD1 inhibitors relative to normal human astrocytes, but there's also some selectivity in terms of upregulation of these immune genes. So what does it mean that these immune genes are upregulated? Is this a way in which immunotherapy could potentially be enhanced? Well, because of the identity of some of these immune genes, we were intrigued by the possibility that this might indeed be the case. And again, Yangshi had shown that LSD1 knockout can actually sensitize cells to T cell cytotoxicity. However, this was done in melanoma models, and when we did in vitro assays to see whether T cell killing was enhanced by these LSD1 inhibitors, we did not see the same trend. So we did this experiment in both DIPG lines as well as in adult GBM lines. And the fact that the DMSO control is roughly the same as pretreatment with any of the LSD1 inhibitors in terms of specific lysis by T cells indicates that there was not an augmentation of T cell induced killing. However, some of those immune genes that were upregulated are also implicated in NK cell killing. And so that prompted us to do NK cell lysis experiments, where we did see that specific lysis of DIPG4 or DIPG6 cells with these LSD1 inhibitors was indeed augmented. And so again, this was primarily with the catalytic inhibitors with phenylcypramine or its cousins, where NK cell killing, again, of DIPG cells is enhanced. This isn't the case for T cell killing of DIPG cells treated with these same inhibitors. We next looked to see whether this was something that could be translated into mouse models for DIPG. And so we were provided the DIPG4 cell lines originally from Dr. Manji and Vidya Gopalakrishnan, who's worked extensively with these lines, labeled them with luciferase, and then also advised us on how to give the NK cells to mice. So we treated these DIPG-bearing mouse models with LSD1 as well as intracranially delivered NK cells. And we saw that the mice that received both NK cells as well as GSK LSD1, which is one of the phenylcypramine backbone catalytic inhibitors of LSD1, actually showed a decrease in tumor burden. So this is this mouse model that we used, the DIPG mouse model is a orthotopic xenograft. So it's not an ideal mouse model in which to study immunomodulatory effects. For that, we really need an immunocompetent model. And we're currently working with an immunocompetent model that was generated by Oren Becker. In that immunocompetent model, we see that treatment with phenylcypramine or GSK LSD1 is actually causing an increase in several of those immune genes. And so we're in the process of understanding how to leverage this increase in immune genes by LSD1 inhibitors for actual translational potential, especially in the context of delivery of NK cells, which, again, the cytotoxicity appears to be enhanced by LSD1 inhibition. So as we're delivering these NK cells to mice that are treated with LSD1 inhibitor, we also have to think about how these LSD1 inhibitors are affecting NK cells. And so we looked at this kind of carefully with our suite of inhibitors, and we came across an interesting finding, which is that LSD1 inhibition is actually compromising the metabolism of NK cells to some degree. And it appears that the scaffolding inhibitors, the catalytic inhibitors less so, but the scaffolding inhibitors in particular have a more dramatic effect on NK cell metabolism. However, we also showed in this recent publication that we can rescue that effect with exogenous glutathione. So if we add an antioxidant, glutathione ethyl ester, the cytotoxicity of the NK cells, which is compromised by the scaffolding inhibitors of LSD1, is rescued. So we have a solution, a potential solution for how to retain NK cell activity and action against a target cell such as DIPG in the setting of using LSD1 inhibitors as an immunomodulatory tool. So in general, we're kind of thinking about the LSD1 inhibitors as a way to enhance immunotherapy, to either prep a tumor cell for immune recognition, but potentially also in modulating the efficacy of maybe not necessarily a T cell, since I showed you that the T cell killing is not enhanced by LSD1 inhibitors, but delivering a modified cell, perhaps a modified NK cell, LSD1 inhibitors could actually increase the ability of the NK cells to kill DIPG cells. So in order to kind of carry this further, we need to understand what is around in the landscape of DIPG cells, what immune cells are kind of populating that area. And so to answer that question, we teamed up with Linghua Wang, who's an assistant professor in the Department of Genomic Sciences, and she was utilizing this exciting informatics pipeline called CyberSort, which basically deconvolutes publicly deposited RNA-seq data, such as the gene set that we originally looked at, LSD1 expression in the immune genes in, to try to define what actual immune cells might be present together with the tumor cells. And so using that technique, she helped us show that activated NK cells are actually conferring a survival benefit in DIPG and pediatric high-grade glioma patient data sets. However, it's important to note that the type of tumors in which this activated NK cell signature shows a survival benefit are ones that are wild type for the histone mutation and are occurring in the hemisphere. And that if you look at significant lymphoid cell infiltration by this CyberSort technique, in general, patients that have the K27M histone mutation compared to wild type patients, and again, these histone mutation bearing patients usually have the tumor incidence in the brainstem, have significantly less lymphoid cell infiltration. So that really highlights the anatomical challenges of delivering immunotherapies and kind of mobilizing immune cells that are present in these patients' tumors, because they're sequestered and they're hard to reach. And so we are excited to explore a role for how LSD1 might make those tumors more attractive to NK cells. And we were gratified to see our paper last year highlighted by a commentary that kind of touched upon some of those aspects of the potential for LSD1 inhibitors to work together with NK cells in eliminating tumors. But we have much to do, which we're in the process of doing. We need to understand, which genes are actually responsible for this LSD1 inhibitor induced enhancement of immune cell killing. And as I mentioned, we're extrapolating this to immune-competent models. And these immune-competent models also provide an opportunity for us to actually look at the neighborhood of these tumors, what immune cells are currently there, and how this seemingly immunologically frigid environment could potentially be thawed. So in DIPG, there's this intense need for improved therapies. And in general, just to pull back a bit to kind of the field of drug delivery or drug development in pediatric oncology, historically, this has been challenging. And so some of the challenges for translating drugs, taking agents like the LSD1 inhibitors and the many agents that have been studied in pediatric oncology before it, forward in clinical trials are kind of exacerbated by the challenges that many of us as folks that are studying therapeutics face in terms of the time it takes to take a drug from the point of drug discovery all the way to even a phase 1 trial, as depicted in this schematic from pharma. There's also the question of which agent to use. For the LSD1 inhibitor story, I mentioned a suite of different LSD1 inhibitors, and they're not all completely equivalent. So choosing the right agent is a key question that has to be answered. If you're using an agent that has been developed by a company, you kind of have to have an alignment with pharma goals, which may or may not be inclusive of pediatric cancer populations, which are typically smaller groups of patients spread across many different sites, again, taking years to recruit patients that will yield statistically significant results in terms of clinical trials. So this time frame and the cost is really even more protracted potentially for pediatric cancer populations. However, there have been some exciting new developments in science policy where pharmaceutical companies are getting vouchers for developing drugs for pediatric populations, which is helping a little bit with kind of this challenge. Frequently, negative trial results in other cancers will shut down a drug forever, and we'll never have an opportunity to actually study a promising drug, which might have been identified as a good agent to use from the science perspective because of negative trial results. So that's another challenge. And then we also have to have markers of whether these drugs are actually hitting their purported target. And this can change from cancer type to cancer type. Frequently, those biomarker studies are not done in pediatric cancer models. So obviously, pediatric cancer therapeutics is important to pursue despite these challenges, but several years ago we started thinking a little bit differently about how we could potentially move the needle on outcomes for pediatric patients by integrating a role for modifiable behaviors. And I just want to make it crystal clear that we're thinking about these modifiable behaviors in conjunction, of course, with therapeutics. But these modifiable behaviors that are highlighted in this cartoon are more frequently studied in the context of cancer prevention and in survivorship, not as much in terms of behaviors that could be kind of leveraged to enhance treatment efficacy. And so energy balance in particular, which is depicted in this schematic, kind of the balance between food intake and energy expenditure, which can lead to changes in weight, has really come to the forefront as one of the modifiable behaviors that can have an impact on treatment efficacy in adult and pediatric cancers. And I wanted to just take a moment to shamelessly plug this upcoming meeting that will be held virtually from July 8th through 10th. That's going to kind of take a deeper dive into energy balance interventions in both mouse models as well as in patient populations. If you're interested in this topic at all, please think about registering. And you can even submit an abstract until May 24th from this link. So one of the challenges for kind of implementing any intervention that addresses modifiable behaviors is when in cancer treatment to give this intervention. And so in pediatric cancer survivors, there have been energy balance interventions delivered, but the survivors are frequently hard to reach. Retention and even reaching out to these survivors is a challenge. We know that in patients, while patients are actually being treated, unhealthy weight gain can actually worsen patient outcomes. And this survival curve is really highlighting that point. This is work from Steve Middleman's group showing that in ALL patients, that subset of patients that usually do very well, if you're overweight or obese, patients have a worse outcome. So the question arises of should we try to address overweight and obesity in patients that are currently on treatment? We know that there are challenges associated with delivering interventions in this setting as well. As you can see from these kids that are enjoying this not so healthy meal, frequently permissive attitudes about diet persist during pediatric cancer treatment, and this is kind of an additional point that we have to think about in delivering interventions. So we took a look back to try to understand kind of the window of time in which it might be optimal to deliver interventions in the patient population seen at the Children's Cancer Hospital. And this is showing the leukemia patients seen over an 11-year period. We did a retrospective chart review of over 200 patients, and about 70 of them were leukemia patients, which were followed from diagnosis into survivorship. And so in this cumulative group of about 70 patients, first off, these patients started out more frequently overweight or obese than we initially appreciated. So almost 50%, 45% of these patients were already overweight or obese. And then as they kind of went through treatment and went into survivorship, the rates of overweight or obesity increased. And this is a trend that has been observed by numerous groups and is certainly present in the population seen at the Children's Cancer Hospital. So we've thought a bit about how to deliver these interventions. This is from a review article written by Carrie Shadler, Dr. Jeannie Kleinerman, the former head of pediatrics, and myself, where we really thought about how not just obesity, the adipocyte interaction with tumor cells, but also the benefits of having a prescribed diet, of having a prescribed exercise plan. So kind of focusing on energy balance without confounding it with the impact of obesity might have benefits. And so this is something that we are interested in from the perspective of diet quality and how diet quality independent of obesity may actually influence outcomes for pediatric cancer patients. And so we actually undertook a randomized counseling intervention. This was in patients that were undergoing treatment for leukemia, but at a point at which they're known to have additional weight gain. So this is during a time frame when these patients are receiving steroids. We conducted a randomized clinical trial where patients were either randomized into an intervention group or a control group. And then on a monthly basis, they interacted with a research dietitian and either got kind of advice that led them towards goals that they had already set for themselves related to healthy diet, or just got education materials in the control group. And we looked at their caloric intake during this time frame over the course of a year, again, with just once a month interactions. And in this patient cohort, which was primarily ALL patients, we saw that the intervention group did reduce caloric intake. So we were able to influence diet simply by having this touch point of once a month through the monthly nutrition counseling. However, we did not change their BMI or their waist circumference. For that, I think we will need to work on both sides of energy balance, exercise, expenditure, as well as perhaps have a more specific type of diet prescribed rather than letting these patients kind of set their own goals in the somewhat loose fashion that we tried in this first intervention. So we've since tried to expand our investigations into trying to really define specific diet parameters that will then guide diet interventions. And then these diet interventions, once they're delivered, we have to find ways to actually maintain these practices. And so this has led to kind of study of all of these domains, defining diet parameters in mouse models, primarily for pediatric leukemias, delivering diet interventions such as the one that I just showed you, and then trying to think of ways to actually sustain these diet changes. So this is an example of some of the mouse work that we've done to try to define what diets might augment chemotherapy efficacy. And here we used a mouse model for acute myelogenous leukemia. And this mouse model bears the FLT3 mutation, which categorizes it as one of the worst types of leukemias in terms of prognosis. These cells were kindly provided by Marina Konopleva, which we put into mice. And you can see that we had fulminant leukemia. When we put these mice on an anthracycline called doxorubicin, which is known to effectively treat this type of leukemia, we saw a reduction in tumor burden as shown in this graph. When we combined a low-fat or low-sugar diet together with doxorubicin, the tumor burden was dramatically decreased. And this was seen with doxorubicin, which is a chemotherapy that's been around for decades, as well as with a targeted therapy, quizartinib, that specifically targets the FLT3 oncogene in this subset of AML patients. So we're in the process of understanding the mechanisms behind this effect of low-fat and low-sugar diet. We know from studies from others that high-fat diets can actually promote AML growth and promote AML progression and resistance to therapy, but the reverse has not been shown. So this begs the question of whether this is something feasible in terms of delivery to the clinic. And so to do that, we've embarked on a study to see whether foods, whole foods that match the low-fat and low-sugar constraints that we had in the mouse chow in terms of percent sucrose, percent fat, can be kind of prepared and delivered to patients while they're on treatment. And so this was done in collaboration with the Bionutrition Research Corps at MD Anderson, which is part of the AIM core facility in the Division of Cancer Prevention. And we had these pediatric patients participate in 2 tasting sessions where they had foods that met these criteria in terms of being low sugar or low fat foods. We had the children give us feedback on whether they would eat the foods. And we also had the parents give us feedback on whether they would prepare the foods and whether they felt that these changes in diet were potentially sustainable. And so along the way, we realized that there was a real need for content around what healthy eating looks like. And so many of the recipes that these children were tasting that aligned with the low fat and low sugar parameters that we see are efficacious in mice are from this cookbook website, which we developed almost 10 years ago now. And so this is the most recent version of the cookbook website. It was updated just a couple of years ago. It's freely available at mdanderson.org/recipes. And the name of the cookbook is the At the Table Cookbook. We have a tab for symptom-related recipes. We have general recipe searches and then tabs for actually trying to make healthy choices in terms of cooking substitutions or healthy snack tips. We also have free access. So this is accessible to anyone, anywhere. It's tablet and smartphone compatible. And there's a lot of content, even in various languages on this website, including brochures in English and Spanish that kind of explain what healthy eating is, what a child should be eating at different ages, consistent with general healthy eating guidelines. And many of these tips, such as the caloric intake information, is available in multiple languages, such as Spanish, Mandarin, and even Arabic. And so all of this content, again, is on this website, mdanderson.org/recipes. And we actually compared the features of this website to other websites that are put forth by academic sources. And so we have 770 recipes in our website, which exceeds the number from these other sources. And then the search options for for finding recipes are somewhat unique in that you can even search by textures of foods, by symptoms. We have a child and family focus as well as videos. And so we're eager to use this website in future interventions and as a way to really promote healthy eating during treatment as well as into survivorship. So much of the work on the website was done together with a really talented graduate student from the School of Public Health, Margaret Raber, who just started her own group at Baylor Children's Nutrition Research Center. And so Maggie was interested in how cooking practices can actually be quantified, because with the website, we are promoting recipes that are healthy, but in general, the field of dietary intake really focuses on the foods consumed, not so much how they're prepared. And so it's obvious to you and I that eating an apple is better than eating apple pie, but dietary recalls may or may not take that into consideration. So the preparation of foods can matter. And so she designed this metric for actually trying to quantify healthy cooking and applied this to families of childhood cancer survivors. And so because this is cooking, this had to kind of be inclusive of the family unit. Maggie actually went into the homes of families that had a childhood cancer survivor and observed cooking behaviors. And what she found was that the cooking behaviors in these families was quite similar to cooking behaviors in families that didn't have a childhood cancer survivor, and not so great in terms of highlighting opportunities for improving both fat and sugar consumption. So there's more to be done in terms of teaching healthy cooking, kind of reinforcing healthy eating. We've tried to set a good example within the Children's Cancer Hospital for kind of cooking and kind of the fun associated with some of these programs. So we've held cooking classes in the patient camps for both our external camps as well as our internal camps. And we've had regular cooking classes pre-COVID on the floor where our pediatric patients are. And we also in 2019 had a event where we brought together local chefs with pediatric cancer patients and had them cook healthy foods together to try to reinforce the fact that cooking healthy can be fun and that healthy foods don't necessarily mean unpalatable foods. So we're continuing our journey to try to define diet parameters. We're looking more closely at specific macronutrients, whether they be amounts of protein, amounts of sucrose, amounts of fat, again in mouse models primarily. We're continuing to look at delivering diet interventions, perhaps employing more stringent parameters such as the low-fat and low-sugar diets that I mentioned. And then with the e-cookbook content, we have an opportunity to deliver the material in the cookbook site in mobile interventions as well as in-person interventions and perhaps in hybrid ways. And this also has, I think, implications for sustaining these healthy diets and cooking habits into survivorship. We're also thinking about how diet modification can be combined with other modifiable behaviors. So exercise is important in terms of kind of working that ratio of energy balance, and we're collaborating with Dr. Jeannie Kleinerman and Carrie Shadler on kind of combining diet modification and exercise modification. We're looking at modifiable behaviors such as smoking. So obviously, we're not delivering cigarette smoke to mice in this way. We have a smoking robot, and this work is being conducted with support from the Department of Epigenetics and Molecular Carcinogenesis, where in leukemia models, we can see that smoking is actually causing AML progression. We're also interested in circadian biology. This is a sleeping mouse, kind of reinforcing the pathways that are important for sleep. Some of these circadian pathways are also involved in diet and exercise induced pathways. And so I just wanted to end the science portion there of today's presentation and go back to the Enjoy Science webinar series. I think many of us have had time to kind of reflect on what we're doing and our purpose. And so I just wanted to share a couple of vignettes about why I enjoy science. And so all of this work is really possible because of the wonderful people we have in our labs. And as George generously mentioned in his introduction, the Graduate School of Biomedical Sciences is a wonderful pipeline for working with scientists in training, and I'm a product of the GSBS here. Fortunate to have several really outstanding graduate students whose work I try to highlight. Mary Figueroa is a current student who's working on the smoking project. Leah Stitzlin is a new graduate student who's working on brain tumor projects. Manu Khosla is working on brain tumor projects and is about to start graduate school. And Megan Rodriguez is currently applying to MD PhD programs. And again, my interactions with the scientists in training are definitely part of what contributes to enjoying science. And also, of course, at MD Anderson, we have amazing multidisciplinary interactions, which I think it's apparent from my talk that I've tried to take advantage of. We're very fortunate to have scientists in so many different disciplines: the Center for Energy Balance in Cancer Prevention and Survivorship, the Center for Cancer Epigenetics, and of course, my colleagues in the Children's Cancer Hospital. In pediatrics, I think we're especially kind of motivated by the potential for patient impact, as I mentioned at the beginning of this presentation. So with that, I'd just like to thank all of the collaborators that have contributed to this work, current and past lab members. We're looking forward to going back to these types of lab interactions versus these types of lab interactions. And we're very grateful to the funding agencies which have enabled all of this work. So with that, I'll end and take any questions.
Thank you very much, Joy. It was wonderful talk, and in fact, it's very, very nice that you bring all this point of diet and the importance much more for the children. Thank you very much. And in fact, also the time remaining is short. I have several questions. So, one question: can the mutation of histone H3 affect the three-dimensional structure and accordingly the global structure of the nucleosome and of the chromatin, doing a global epigenetic regulation alteration?
Yeah, I think that's a great question, and I think it can. I don't think the X-ray crystallography studies per se have been done as yet for actually looking at the complex the complexes coming together, but there's certainly major influences on the PRC complexes, for example, by these histone mutations. So complexes that are controlling chromatin occupancy are affected by the histone mutations.
Thank you. Now another question, more technical, from a curious GSBS student. How was the low sugar, low fat diet monitored in mice?
How? Yeah, so we're in the process of understanding that. We think that it's involving several mechanisms. Oxidative stress seems to play a role, but perhaps oxidative stress in specific subtypes, perhaps in macrophages, and perhaps through modulation of some of the circadian proteins that I alluded to at the end. And so these are active experiments that we're undertaking with Kristen Mahan, who's a circadian biologist at UT Health. And I'll hope to tell you more about that in the future. We'll have a vignette about that in the upcoming energy balance meeting.
And one more question. So did you check in this type of studies what's happened with the non-coding part of DNA? I don't speak about RNA. On the genome, no, the large non-coding regions of the genome that are very important.
Yeah, I think that's a great question. And so if you're talking about the LSD1 inhibitor story, we have gone back to look at some of the long non-coding RNAs that are regulated by LSD1 knockdown. And there's some very interesting pathways that those long non-coding RNAs are associated with. We haven't done that degree of investigation into our diet studies, but I think I'm sure there's going to be a lot of data to be gained from doing those sorts of experiments. We'd love to collaborate if that's something you're interested in. We can think of creative ways to fund those studies.
Thank you. Thank you. Okay, so thank you very much, Jaya. Your talk was wonderful. Thank you for being with us here. And of course, thank you to all the attendees. And in fact, enjoy science. Look, it pop up one more. Okay, let me talk because it pop up right now. I ask the question. How did the immune genes which were affected by LSD inhibitor treatment align with cyber sort analysis? Was there a specific cell type affected?
So we didn't look that carefully in the cybersort analysis. We basically looked at more broadly what immune genes were associated with different subtypes of pediatric high-grade gliomas. But that's an excellent question, and I think one that is addressable with the cybersort platform.
Thank you very much. Thank you. And also I want to make one note from my side to the attendees. So I'm sorry by not saying your names when I say the question, but this was our rule when we start Enjoy Science. And to be fair with everybody who asked questions, of course, I can say only thank you guys for asking the question. Sorry for not naming your names and coming back. Thank you, Joya, for being with us. And thank you all of you. Enjoy science, make discoveries, and let's be happy because science, it's a hobby. And what we are doing, it's changing the world. Thank you again. Thank you, Joya, and thank you all of you.
Thank you, George, and thank you to everyone for participating. If anyone wants to email me, I see this question about the 3D structure in the chat. I'm happy to dive into that more in emails. So thank you.
Thank you all. Take care. Good luck.
Bye, you too.